students, welcome back to Applied Environmental Microbiology. Today we are going to see a very interesting and promising method to utilize whatever we have learned so far in Applied Environmental Microbiology course so that we can fulfill our very important need to be able to detect contaminants, pathogens and other risk agents in our environment faster and with higher reliability. So, what we are going to talk about today are biosensors. Now, sensors, as the word in itself tells you, is something that you use to sense or pick up data, either quantitative, semi-quantitative or even qualitative. Now, we must be very careful about this particular topic because while a lot of research is currently underway to develop better biosensors, better sensors, there is a dearth of sensors that can be used at low cost for robust and reliable measurement of environmental contaminants. Now, in a country like India or in any other developing country, we have the problem of overpopulation and pollution. As a result, we don't even know what kind of contaminants, what kind of pollutants are present in our water, in our air and in our soil and definitely on our food. And not only environmental contaminants, but also pathogens are a big challenge in a developing highly populated country such as India, because somehow Indian subcontinent is the perfect hotbed for emergence of antimicrobial resistance. As a result, the pathogens that we have in India might have, or in anywhere else in the world actually, might have higher levels or elevated levels of antimicrobial resistance. So, it is not only important, not only sufficient for us to know whether a particular pathogen is present in the sample or not, but also to find out rapidly whether the pathogen, if present, is resistant or not. If you remember what we studied in our previous lectures on antimicrobial resistance, one of the key themes was that it is not just important for us to pinpoint the pathogens that have the ability to resist antimicrobials, but we should be aware and alert about all kinds of microbes that have the potential to display antimicrobial resistance genes. So, for us we have emerging contaminants, such as AMR, antimicrobial resistance and antimicrobial resistance genes. Then we also have highly contaminated environment. And now this is coupled with high demand on resources. And public health burden. So, think about it, dear students. In our country, we have a highly contaminated environment. So, it is very important for us to be very cautious and proactive to ensure public safety and environmental health. Next is, we also have very high demand on our resources. So, we only have limited amount of resources and we have to make sure that we use all that we have to its optimum, and as such we cannot afford to waste our resources. The other thing is we have a very high public health burden from infectious diseases. As a result, it is important we need urgent data collection. So, rapid data collection is required. So, we need rapid, reliable and robust data collection methods. So, basically this is the need of the hour today in our country, rapid, reliable and robust detection techniques for environmental contaminants. As such, biosensors are a promising avenue, and this is beautiful part of biosensors, that not only do they help us detect our environmental contaminants, but also they use the biology, our understanding of biology, to create very specific, very sensitive and reliable sensors. For example, we know that if you go back to our lectures on central dogma, where I talked about genes, how they translate into proteins and how they encode for all cellular activities. So, in this you would remember that genes they follow a very strict sequence pattern which has some leeway here and therefore degeneracy, but mostly a reliable strict sequence pattern. Now we can create such very specific sequence patterns that will fold or that will behave in ways that will only react, respond or give a signal when it attaches to a target. As such, they hold potential to become biosensors. Same is true with proteins and same is true with antibodies and many other biological entities that can be used, that are being used to make biosensors. So let's progress, let's go ahead and study more about biosensors. Now what is biosensor? Now this is a definition that has been taken from IUPAC 1996 Standard Journal. It says a biosensor is a self-contained integrated device that is capable of providing specific, quantitative or semi-quantitative analytical information using a biological recognition element which is in direct spatial contact with a transduction element. Alrighty, let's break this down, break this definition down. Basically, biosensor is self-contained integrated device. It is self-contained, it is complete in itself. We do not need to attach it to some other instrument. It is enough for, it is complete in itself. It is integrated device. So, all different parts of any sensor, which is a receptor, a transducer and a signal receiver, all of them are integrated in one device. It is capable of providing specific. This is very important word. Now, the word specific here says that we have some target compounds that we are interested in. For example, if I ask you the question, is the water dirty? Is the water, if you give me a glass of water and I ask you, is the water dirty? It is a very generic question because you might say counter ask me a question saying, well Gargi, what do you mean? Do you mean does it have microbes, does it have dust, does it have something that will harm you? Do you mean biological agents, chemical agents, or physical properties are not good enough? What are you looking for? Do you want 100% pure water, or what some impurities are okay, what impurities are not okay? So there are many questions, follow-up questions that we can ask to clarify the very generic question of is the water clean or is the water dirty. However, biosensors or any sensor, especially biosensors, are helpful when we are very targeted in our questioning, not very generic. For example, if I ask you or if you ask me that, hey, are there antimicrobial resistant genes in this water? And then we might even go ahead and say, well, I am interested in methicillin resistant Staphylococcus aureus. Are they present in the water? Or are the genes that confer methicillin resistance, are they present in the water? So this is a very targeted question, and thus the compounds that I am looking for are very targeted. Which brings me to another point, we will cover it later, but let us hear about it anyway. When you are targeting particular compounds or particular set of compounds, we are very interested in specificity. So specificity basically says that your sensor will give a positive signal only when the target is present. Let us say I have MRSA gene. Now MRSA is a gene that confers resistance to methicillin antibiotic. So any microbe that has MRSA and it is actively translating it, actively expressing proteins related to MRSA gene, then that particular microbe will be resistant to methicillin. Let us say I am also interested in vancomycin resistance, okay, and let us say I know that there is another form of resistance present in my water, alright. So let us say I have drinking water, a glass of drinking water, and I want to find out whether MRSA is present in it, whether VANA is present in it, because these genes are marker for methicillin resistance and vancomycin resistance. Now let us say my biosensor or my sensor gives me a positive signal, yes, your target is present. So it is quite possible that it is giving me signal for MRSA or it is giving me signal for VANA. But if it also gives me signal for BLA NDM1 when I had only designed it to give me signal for MRSA or VANA, then this is not specific. It is and let us say there is another glass of water, okay, another glass of water, and let us say in this we only have NDM1 resistance genes. So there is genes that give resistance to beta lactamase. Now and this is New Delhi metallo beta lactamase, by the way. So only this particular genes are present in this water, and if I put my sensor in and it gives me a positive signal, this is not specific, because even when the target and MRSA and VAN-A were not present, it gave me a signal. So it is very important that my biosensor only gives me signal when my target compounds are present. So that is what the specific word is here. Now quantitative or semi-quantitative analytical information. Ideally, my sensor should not only tell me if MRSA or VAN-A are present or not, but it should also be able to tell me how many MRSA copies are present, how many VAN-A copies are present. So for example, if I have 10 to power 7 per ml copies of MRSA and 10 to power 2 per ml copies of gene copies of VAN-A, then my sensor should be able to give me an estimate for this. Sometimes it is not quantitative per se, but it is semi-quantitative. Now what is semi-quantitative? Let's just try to understand that. Now in semi-quantitative, my sensor may not be able to tell me exact value, how much per ml of MRSA gene copies are present and how many VAN-A gene copies are present, but it might be able to give me an idea like this, an idea such that the ratio of MRSA gene copies to 16S rRNA gene copies is something. Now this is semi-quantitative, it is giving me relative abundance, or maybe it is giving me an estimate of which is more, the MRSA is more or 16S rRNA is more. Well, let us take another example. So let us take another example. Let us say it is giving me an idea of, if let us say it is semi-quantitative, and the sensor is giving me an idea of MRSA ratio VAN-A, and it says 4:10. Now because it is semi-quantitative, I cannot say that, well, I have 2.5 as many copies of VAN-A genes as MRSA, but what we can say that perhaps VAN-A is more, at least twice as much as MRSA. So this is because it is semi-quantitative, we cannot rely on these numbers completely, but it does give us an idea of the relative abundance, it does give us an idea of the trends. So if over time the MRSA decreases or VAN-A increases, I know the trend. So that is the importance of semi-quantitative, and because it is a biosensor, it uses a biological recognition element. Now how would it do that? For example, we know a particular sequence for MRSA, we have already sequenced it, we have also sequenced VAN-A. So one very simple technique could be, we have an aptamer polynucleotide with the sequence that is perfect complement to MRSA, and somehow we can make it specific to MRSA, and when if it attaches to MRSA, if it does, we need to do a lot of troubleshooting for that. So that is a part of research, and if they complement perfectly, and then after this pairing it should give me a signal, and that signal should be read by my detector. If I can do this, then I have my sensor ready, biosensor ready. Alrighty, so now let us go ahead, and the biological recognition element is in direct spatial contact with the transduction element. So the recognition element gives signal via transduction element, and they are in physical contact with each other. So this is your typical biosensor. Here is your analyte, which is another way of saying your target, whatever contaminant or whatever you are interested in. Here is your receptor, bioreceptor. So this is your bio element, and because this is biosensor, it got to be bio elements. It can be protein, it can be an antibody, an enzyme, it can be some genetic material like RNA based aptamer or DNA based aptamer. And then you have transducer. This is very important. So what transducer does is, when analyte, when the target interacts with the bio element, either they aggregate or they behave in particular peculiar ways, then transducer creates a signal. It can be an electrical signal, it can be an optical signal, and as we will go for, it can be some other kinds, conductivity based signal and other kinds of signal, and then the signal is read, and when the signal is read and processed, you get information. Yes, target is present, no, target is not present, and if target is present, we want to know how much target is present, or at least get some semi-quantitative information. Alrighty. So briefly, we can say a biosensor is any device that uses specific biochemical reactions to detect target, and this is one particular example, which is mutagen monitor. So here you have your microbial cell, here you are shining laser light, blue light, and the ones, if the cell has some kind of mutation in it, the kind of mutation you are looking for, carcinogenic mutation, then it will emit light of different color. The blue will appear to as green. So, because your sensors are actually jellyfish gene produces fluorescent protein whenever DNA gene repair is activated. So, you are using this gene from jellyfish, and so why would DNA repair gene be activated? Because there is damage in the DNA. So, the way the mutagen monitor works is that there is DNA, it gets damaged, and when DNA gets damaged, what it does is it activates repair genes, because the damage could be deadly for the cell or not helpful for the cell. So, it activates repair genes, and the repair genes, when they contact with or when they interact with a particular gene from jellyfish, let us just call it jellyfish gene, then together they are fluorescent, they produce a fluorescent protein. So, when they interact with each other, the protein that will be expressed will be a fluorescent protein. Now, this fluorescent protein will change the light of the color of the light, and we will be able to tell whether repair genes were triggered or not, whether they were active or not. So, this is how we can detect mutagenesis in cells, and this is a biosensor, because we are using biological agents for detecting cancer or mutagenesis. Now, the current definition of biosensor is a sensor that integrates a biological element with a physico-chemical transducer to produce an electronic signal proportional to a single analyte. So, this is our attempt to get quantitative information. Alrighty. So, sensor integrates biological agents. So, there has to be a biological agent, we can call it bioreceptor, and it is in contact with a transducer which is physico-chemical, and they are saying it produces an electronic signal. Notice, not electrical signal, but electronic signal, something that can be read by our computers, and the signal should be proportional to a single analyte. Basically, if you have one target, you get, let us say, X amount of signal. You get X amount of signal. So, if you have 10, you should get 10x amount of signal. If you have 100 targets, you should get 100 this much amount of signal, 100 times more signal. So, it becomes quantitative, and then this is conveyed to the detector which feeds to a computer, and we can read data. Alrighty. So, basically we can say that biosensors are bioreceptors plus transducers. So, there is a receptor, and this is bio, and this is the part of biosensors that give the bio part to the biosensors. So, these are the biological agent. Transducers are basically physico-chemical, and they can be anything. You can have proteins, enzymes, you can have DNA, genetic material, RNA, polynucleotides, anything. The sky is the limit here, and then there is a transducer which converts the interaction between bioreceptor and the analyte into an electronic signal. So, transducer is very, very important. Now, as an example, enzymes are bioreceptors. Now, if you go back to our previous lectures, you would remember that we talked about what enzymes do. Basically, as a revision, enzymes catalyze reactions in our body, that in a microbial cell, also in a cell of a big organism such as human beings. What they do is they catalyze the reactions that would not happen otherwise at room temperature. So, they reduce the overall energy, and they allow an easy jump over the reaction and energy barrier. And how do they do that? For example, very simple question, when we eat food and we taste our food, we start salivating. So, in this process, tasting the food, salivating, or thinking about food and salivating, there are certain enzymes that are at work. And these enzymes, what they do is they interact with your food, and they know what kind of food it is. So, they will inform your body, okay, need to produce more insulin or less insulin, produce more of this enzyme, less of this enzyme. Now, and then the insulin enzyme will interact with the glucose in your blood. Now, regardless of which enzyme is working where, there is one thing that is common to all enzymes, that enzymes are very specific. For example, if there are amino acids floating in your blood after having a good hearty meal, insulin will not react with them. It is very specific. But if there is glucose in the blood, then the insulin will interact and do its job, what it does. Thus, the enzymes are very specific. And how come they be specific? how does an enzyme, a protein, know who it is interacting with? In order to know that, for protein, the structure of protein behaves like a biosensor. Let's say this is your enzyme, and let's say insulin hormone, and now here you have molecules of sugar. So it so happens that they too fit like lock and key, and when they fit, they come in a perfect shape. Whatever that shape may be, I do not know, I am just making a schematic here, but in this perfect shape, now insulin can do its job, right? But if it interacts with other things like amino acids, no change in configuration, no change in conformation. Thus enzymes behave like bioreceptors, and no wonder one of our first biosensors were based on proteins, were based on antibodies and antigen antibody interaction, were based on enzymes, enzymatic activity. So if I have the right enzyme, I know it is specific, it is the chemistry and the physical properties of the enzyme are specific to a particular target. So if I want to detect the target, let us say I want to detect glucose, I can take enzymes that specifically interact with glucose, because they know they change their conformation, they change their structure and their qualities once they have interacted with glucose. So they are perfect bioreceptors. And to make a biosensor or bioreceptor, let us say I have chosen a bioreceptor, some enzyme E, and I know this enzyme E interacts with target T. Target T is what I am interested in, because it is harmful when it is in the environment. When it is in the body, I know that enzyme E acts with target T. So what I need to do is I need to have a way of physically entrapping, immobilizing the enzyme on the surface of my sensor. So this is my sensor that I am making, biosensor. So I need to find a way through which I can make sure that the enzyme E attaches to the surface in which I am going to put the sample. So this is one of the first challenges that comes when we make biosensor, which is immobilization of bioreceptor. It can be done in two ways. One is physical entrapment, we physically force the enzymes to be stuck here. The second is chemical attachment, so they can be attached through a chemical bond. Now next is transducer. Now basically once the enzyme, and this is your analyte, okay, the analyte that target T. So once they interact, they undergo a change. So what was E and T separate will now look like this. So this is still E, these are still E, but this particular E has interacted with T, so it becomes something else. And now when this becomes something else, there goes a signal. There is some change, whether it is change in structure, change in chemistry, change in pH, change in conductivity, change in the optical properties. Now this is converted into an electronic signal by a transducer. All these changes in any one important property is translated by transducer. So an example would be, if I am looking at oxidation of glucose, I am very interested in seeing microbial oxidation of glucose. I have glucose and I know there is oxygen reaction happening, and I make gluconic acid and hydrogen peroxide. I have three options. So once the reaction has happened, glucose has reacted with oxygen, I have changed the property of this particular enzyme. It has interacted with the target, with the analyte. So now this can be read, the various properties of this changed conformation of G that can be read and can be converted into an electronic signal. For example, again oxidation of glucose forming gluconic acid and hydrogen peroxide. I have three options here. I can have a transducer here whose job is to read the amount of oxygen available in environment. So as oxygen drops, it knows that glucose oxidation is happening. So it can detect oxygen, or it can detect gluconic acid. So as gluconic acid is present more and more, transducer creates more and more signal. So we know now the reaction is moving towards the right side. Or we can have hydrogen peroxide detector. So all these three things are subject for detection, and either one of them will give me information on oxidation of glucose. Okay, now let us look at components of biosensor in little bit more detail. So these are your enzymes. Now coming back to the whole enzyme business, enzyme, antibody, microorganism or capacitors, and now these are very specific to a particular target. So in your sample you might have different kinds of compounds present. So we have the green squares, red pies and blue circles. Now all of these will interact with the enzyme, but only the red pies would fit perfectly into the enzyme, and once they fit perfectly, it undergoes a conformation change. Now this could be a change in electroactive substance, it could be change in pH, in heat, light, even mass, because it might release some other thing away or gain some mass obviously. And either of this can be detected and converted into signal using a transducer, which can behave like an electrode, like a semiconductor or pH electrode, thermistor for heat, and for light a photon counter, for mass a piezoelectric device, and so on and so forth. This is the job of transducer. And then the transducer has converted this conformation change into measurable signal, and then you can read the electronic or electrical signal. Now let us look at some of the bioreceptors. We have enzyme, we have talked about it. Antibodies, remember antibodies are part of human defense system. So antibodies are very specifically complementary to antigens. So antibody A will be specific to antigen A. So if you want to detect antigen A, immobilize antibody A on the surface, bring in contact with the sample. Whenever it will interact with antigen A, it will undergo conformational change, have a nice transducer which can translate this into an electronic signal, and you have detected antigen A. Alrighty, then we have microorganisms. There are certain microorganisms that also behave well as bioreceptor, and then we have CDL. As transducer, I have already talked about these things, so let us move on. This is bioreceptor, this is transducer, and the electrical signal will go to your detector, which will convert it into data. Alrighty. Now what are the requirements for bioreceptors? We have so many options. We can use enzymes, we can use aptamers, polynucleotides, we can use DNA based, RNA based bioreceptors, we can use antibodies based bioreceptor, microbial bioreceptors. So the requirement for bioreceptor would be, we need to select a suitable bioreceptor molecule, right, which is specific, sensitive, and we will talk more about this. We need to also select a suitable immobilization method. Alright, I have found the best aptamer for detecting a contaminant I am very interested in, but how do I immobilize this this receptor without damaging its business, damaging its function? Next is, I need to select a suitable transducer. If I know that one particular contaminant fits perfect lock and key with a particular bioreceptor, I need to find out, okay, what would be the changes in their properties, and what transducer would be able to convert this change in properties into an electrical signal. Then I need to design biosensor considering measurement range, linearity, and minimization of interference. These are very important. Many people have already crossed the first, second, third barriers, but the fourth barrier is, what is my measurement range? What is my sensitivity? For example, if I tell you I can make a sensor, biosensor, that will give you values of 10 to power 8 gene copies of antimicrobial resistant genes per ml, and this is the lowest limit it can detect and above. This might not be very important and very valid in developed countries such as Britain, which are very clean and have in general lower levels of AMR. And also in our country, we know that the public health threats even of 10 to power 3 to 10 to power 4 gene copies per ml of AMR genes can be tricky and dangerous. So measurement range is very important. This is not very useful if it is higher than what I am interested in. Next is linearity. So remember, in previous slide I was telling you that if I have one analyte present, this is analyte by the way, then I get X signal. If I have 10 analyte, I get 10X signal. If I have 100 analyte, I get 100X signal. After a time, what we notice is that the linearity goes away. See, for 10 to power 5 analyte, I only got 4000 X signal, not 10 to power 5 X signal. So this is the vanishing of linearity, and this limits my upper range and also my lower range. So this is also very important, measurement range and minimization of interference. So in lab, when we make our biosensors, or in human body also when we have our enzyme doing their job, the human body is arranged in such a way, and other life forms are arranged in such a way that the enzyme that is specific to a particular analyte will find each other more often than not, otherwise the life can't continue. So, and they won't find other kind of crazy contaminants that might interfere with the lock and key perfect fit mechanism between enzyme and analyte. But in environment, this interference business is very, very important. For example, you might have heavy metals that might completely shatter your bioreceptor, and then bioreceptor doesn't work. because of interference, or there might be other contaminants present in the water, or in the soil, in the sample, in the food, for example humic acid and other natural organic matter that will interact with your biosensor, bioreceptor, and then you will not be able to get a good reliable data. So we want to minimize the interference. Next is we want to package the biosensor in such a way that it is compact, robust, definitely cost effective, and that we can use over and over again. So the basic characteristics of biosensor should be linearity. So as the concentration goes high, so this is concentration on x-axis and this is the signal on y-axis. So ideally it should be a straight line, but as the concentration goes high it plateaus. So this should not be there, should have a very nice, very reliable linear range. The other is it should be very sensitive, which means I should be able to detect lower and lower values. Next is should be very selective, means very specific. So basically I do not get false positive. And the response time should be time necessary for having 95% of the response. So the response time is very important. Again, in the beginning of this lecture I mentioned that how it is very crucial, very important that the whatever biosensors or whatever detection techniques we use to detect contaminants in our environment, we can use them rapidly, because we have a crunch of resources and we have a severe public health burden. So we cannot afford the delays. That is the importance of biosensors also, because some detections, for example tuberculosis detection, mycobacteria might take really long time in the lab to grow, and by the time we get a positive result from the lab the patient might suffer a lot. So quick detection techniques are very important and required. So dear students, this is all for today's lecture. In the next lecture we will explore more about biosensors, we will even explore different kinds of biosensors. Thank you.